At depths where sunlight never reaches, evolution repeats a chilling pattern. In every era, in every trench, the darkness breeds giants, colossal squid, monstrous isopods, amphipods the size of rats. Modern science tracks their growth, but the true explanation lies deeper than rare food or ancient age. Why does evolution create the same massive life forms again and again, in sterile black oceans that seem almost timeless? The answer begins where cold and pressure push biology to its breaking point. In the deep, cold reaches of the ocean, time itself seems to slow. Here, the water hovers between 2 and 4 degrees Celsius, barely above freezing. Life moves at a different pace, and the rules that shape bodies on land or in sunlit shallows no longer apply. Instead, cold rewrites the script for survival, stretching out lifespans and transforming the very nature of growth. The Greenland shark drifts through these icy depths, almost a living shadow. Its flesh is cold to the touch, its heartbeat slow and steady, its movements unhurried. Scientists have traced the age of these sharks using radiocarbon dating of eye lens proteins, revealing individuals that lived for centuries. Some Greenland sharks do not reach maturity until they are over 150 years old. This is not an outlier, but a pattern written into the biology of deep-sea giants. In cold water, metabolism drops to a crawl. Each heartbeat, each breath, each cell's work is stretched out over longer intervals. Energy is precious and spent sparingly. For many deep-sea animals, growth never truly stops. Unlike most surface dwellers, they are indeterminate growers, adding new tissue slowly, year after year, as long as they live. The cold grants them this gift, a longer window for growth, a slower pace of life that opens the door to greater size. It is a twist on Bergmann's rule, which says that animals in colder environments tend to be larger. In the deep, the effect is magnified. The chill at depth does not just slow the clock, it extends it, turning decades into centuries. The Greenland shark is joined by other slow giants, colossal squid, giant amphipods, Antarctic sea spiders, all following the same quiet logic. Their bodies grow large not through a rush of abundance, but by accumulating mass over vast spans of time. In these frigid waters, time itself becomes the currency of growth. The longer an organism can endure, the larger it can become. But cold alone is not the only force at work. The deep sea's oxygen story will add another layer to this puzzle, shaping who can grow and how long they can last. For now, in the silent dark, the slow heartbeat of a Greenland shark is a reminder. In the deep, survival means trading speed for endurance and letting the years do the work of giants. Dissolved oxygen carves invisible boundaries through the ocean's depths. Near the surface, wind and waves churn oxygen into the water, sustaining a riot of life. But as sunlight fades and organic matter drifts downward, a silent transformation begins. Between 200 and 1,000 meters, microbial decomposition of sinking debris strips oxygen from the water, creating the oxygen minimum zone. Here, oxygen drops to some of the lowest levels found anywhere in the open ocean. Many animals avoid these midwater deserts, or survive only by slowing their activity to a crawl. Below this suffocating belt, a paradox unfolds. Past the oxygen minimum, as the water sinks deeper into the abyss, oxygen levels begin to rise again. Cold polar currents, rich in dissolved oxygen, plunge along the seafloor and spread through the world's great trenches and plains. At depths of 4,000 meters and beyond, the abyssal ocean is often more oxygenated than the shadowed waters above it. This reversal is not intuitive, but it is fundamental to the existence of giants in the deep. The chemistry of the deep sea is shaped by slow, relentless mixing. Antarctic bottom water and North Atlantic. Deep water, two massive oxygen-laden currents, sink in polar regions and creep along the ocean's floor for thousands of kilometers. Their journey takes centuries, but they carry enough oxygen to support life even in the world's most remote trenches. In places like the Mariana Trench, oxygen concentrations at 10,000 meters can rival or exceed those at the surface. The deep is not a stagnant wasteland, but an environment where oxygen is quietly abundant, waiting for creatures adapted to use it. This abundance is a gift for the giants. Large bodies need more oxygen to power their cells, even if their metabolism is slow. In shallow or mid-water zones, limited oxygen can choke off growth, forcing animals to remain small or restrict their activity. But in the abyss, where cold, oxygen-rich water flows across the seafloor, the rules change. Here, a giant isopod or amphipod can support slow, steady aerobic metabolism, growing larger year after year. The oxygen paradox, scarcity above and abundance below, creates a hidden refuge for the evolution of size. The deep ocean's oxygen profile is not uniform. Local currents, geography, and ancient climate patterns shape how much oxygen reaches each trench or plain. But wherever deep currents flow, they bring the possibility of life on a scale impossible in starved midwaters. Oxygen, invisible and silent, becomes the second pillar of deep-sea gigantism. It is the chemical breath that fills the bodies of giants, sustaining them through centuries of darkness and slow growth. At the molecular scale, life in the deep ocean is a constant battle against crushing force. Every 10 meters of depth adds another atmosphere of pressure. By 7,000 meters, the weight of water above is nearly 1,000 times what surface creatures experience. Under these conditions, the fragile architecture of proteins, their folds and twists that make life possible, begins to unravel. Without a defense, enzymes lose their shape, membranes stiffen, and the machinery of the cell collapses. Yet deep-sea giants endure, their cells braced by a simple molecule, trimethylamine N-oxide, known as TMAO. This compound was first isolated in the tissues of abyssal fish, and it acts as a molecular shield. As pressure rises, TMAO accumulates in muscle and blood, weaving itself into the watery environment that surrounds every protein. Its effect is subtle but profound. TMAO strengthens the hydrogen bonds between water molecules, making the liquid matrix less compressible and less prone to collapse. With this invisible scaffolding in place, proteins can keep their shape even as the world around them is squeezed from all sides. William C. Yancey, a pioneer in the study of pressure adaptation. In his words, it is the molecule that lets life push deeper step by step into the abyss. Yancey's research revealed a simple rule: the deeper a fish lives, the more TMAO it carries. This relationship is so precise that you can estimate an animal's depth simply by measuring the concentration of TMAO in its tissues. At shallow depths, TMAO is barely detectable. In the muscle of a snailfish from the Mariana Trench, it reaches levels that would be toxic to most surface dwellers. TMAO's power lies in its ability to anchor water's structure, which in turn stabilizes the delicate folds of proteins. Under pressure, water molecules are forced closer together, distorting the hydrogen bonds that hold them in place. TMAO counters this by reinforcing the bonds and preserving the environment proteins need to function. Without it, even the simplest reactions, breaking down food, copying DNA, moving a fin, would grind to a halt. This molecular defense, elegant in its simplicity, sets a ceiling on how deep complex life can go. As TMAO builds up, it creates new challenges for the cell, forcing organisms to balance the benefits of pressure, resistance against the costs of rising osmotic stress. It is the unseen brace that lets their bodies withstand the weight of an entire ocean. Far below the reach of sunlight, another boundary rises, one set not by temperature or oxygen, but by chemistry. The deeper a creature lives, the more it must invest in survival at the molecular level. For every 10 meters descended, the ocean presses harder, and the cost of withstanding that pressure increases. The deep sea's most reliable defense is trimethylamine N-oxide, known as TMAO, a molecule that braces proteins against being crushed. Yet this solution comes with a price. As TMAO builds up in tissues, it draws in water and raises internal osmotic pressure. Cells must spend energy to keep their chemistry balanced, and the deeper they go, the steeper the cost becomes. At around 8,200 meters, this trade-off reaches a breaking point. If a fish were to descend further, it would become isosmotic, its internal fluids matching the saltiness of the ocean. At this threshold, the machinery of life falters. Water and ions can no longer be regulated, and the cell's delicate balance collapses. This is the isosmotic barrier, a hard chemical wall that fish cannot cross. No known fish has ever been found deeper. The Mariana snailfish, Pseudoliparis swirae, lives at the edge of this frontier. In the Challenger Deep, it drifts through water at nearly 11,000 meters, but it never strays far from the upper Hadal zone. Every cell in its body is finely tuned to survive in a world where pressure would unravel the proteins of any surface dweller in seconds. Yet even the snailfish cannot push much deeper. The chemistry would no longer work. For fish, the abyss has a bottom not set by rock, but by molecules. Other creatures, amphipods, isopods, and certain invertebrates have found different solutions. They use alternative osmolytes or evolve even softer bodies. But for vertebrate giants, the isosmotic threshold is a final limit. The deep ocean is vast, but its chemical laws are absolute. Evolution's reach is long, but here it cannot stretch any further. In the darkness below, 8,200 meters, the giants of the deep meet the edge of what life's chemistry can afford. When a whale dies and sinks to the ocean floor, the deep sea receives a rare windfall. The carcass, weighing up to 50 tons, becomes the center of a slow-motion ecological drama. The first arrivals are the giants of the abyss, giant isopods, amphipods, and hagfish, drawn from kilometers away by faint chemical plumes drifting through the water. Time-lapse studies by Dr. Craig R. Smith and his team have mapped the sequence. Within hours to days, the mobile scavenger stage begins. Giant isopods, some nearly half a meter long, swarm the carcass, tearing into soft tissues with armored jaws. Amphipods, 10 to 20 centimeters in length, crowd in waves, their bodies glistening in the camera-red light. These scavengers gorge until their abdomens swell grotesquely, sometimes immobilizing them for days. The feast is brief, a matter of days or weeks, yet the energy stored can sustain them through years of famine. As soft tissue vanishes, the opportunist phase takes hold. Polycate worms and smaller crustaceans colonize the remains, burrowing into the sediment and feeding on scraps left behind. The scavenger giants retreat, their bodies heavy with stored fat. Over months, the whale skeleton is gradually stripped, and the energy pulse radiates outward, supporting a ring of life in the otherwise barren plain. Months pass, and a new community takes over. Sulfide oxidizing bacteria form mats across the bones, converting the whale lipids into hydrogen sulfide. This chemical energy supports dense colonies of clams, mussels, and more worms, each adapted to thrive on the toxic byproducts of decay. The bacterial mat phase can last for years, extending the whale influence far beyond its death. In the final act, the bones themselves become a resource. Specialized bone-eating worms, Osedax, bore into the skeleton, extracting the last reserves of fat and protein. Over decades, the whale is reduced to fragments, and the site slowly fades back into the deep silence. The entire succession, scavenger wave, opportunist bloom, bacterial mat, and bone reef, can stretch for decades. Each phase supports different giants, each arrival timed to the changing chemistry and structure of the carcass. Dr. Smith's time-lapse deployments in Monterey Canyon have captured these transitions in detail, showing that a single whale fall can support life for up to 70 months, with chemical signatures lingering in the sediment for even longer. The scale of these events, how a single body can feed thousands, and how giants gather in the dark, reveals the deep ocean dependence on rare, unpredictable feasts. Here, size is not just an adaptation, it is a strategy for survival in a world defined by scarcity and sudden abundance. When a giant isopod finds a whale fall, the advantage of size becomes immediately clear. It plunges into the feast, devouring soft tissue until its armored abdomen swells to the point of immobility. This is not gluttony, it is adaptation. In the deep sea, food is so rare that a single meal must last months, sometimes years. Laboratory studies have documented giant isopods surviving 5 years without another bite. Their bodies are built for this, thick with lipid reserves, organs adapted to store energy for the long famine ahead. This storage capacity is not just a matter of volume. Lipid-rich tissues act as fuel tanks, filling with energy during the rare moments of abundance. A large isopod or amphipod can gorge far beyond what a smaller animal could hold, packing away calories that will be slowly burned as the carcass is stripped and the plain returns to silence. These reserves are not only for survival, they are for movement. In a world where the next meal might be kilometers away, a bigger body means a wider range. Larger scavengers can travel farther in search of the chemical trails that signal fresh carrion, increasing their odds of finding the next feast before starvation sets in. The advantage extends to reproduction. Larger females can produce more and bigger eggs, each packed with yolk to fuel the next generation through the long, hungry drift before they find their own food source. In the deep, where encounters between individuals are rare and young must fend for themselves. Energy storage also shapes behavior. After feeding, giant isopods and amphipods retreat from the carcass, heavy and vulnerable. They move slowly, conserving every calorie. Their metabolism drops even further, allowing them to stretch their reserves as long as possible. The feast is brief, and the famine is endless. Only those with the capacity to endure both can persist in the deep. This logic repeats across species and eras. Whether it is a giant amphipod at a whale fall, a colossal squid lurking in the mid-water, or an ancient ostracod sifting through Paleozoic mud, the rules are the same. Size is not just a consequence of slow growth or cold water. It is a tool for survival, forged by the unpredictable rhythm of feast and famine. In the deep sea, to be big is to be prepared, to store the memory of abundance against the certainty of hunger. Long before any human set eyes on the ocean depths, giants ruled its shadows. Their remains etched into stone and pressed into ancient seabeds hint at a world where size meant survival. In the Paleozoic era, about 450 million years ago, trilobites as long as a human arm and eurypterids, the so-called sea scorpions, stretching over 2 meters, prowled the cold, oxygen-rich waters. Their segmented, armored bodies patrolled the continental shelves, thriving in an Earth still reeling from extinction aftershocks. These creatures were both predators and prey, their size a response to the pressures of a world in flux. As the ages shifted into the Mesozoic, the faces of the giants changed, but the pattern held. The end Permian extinction cleared the seas, and in the Triassic ichthyosaurs appeared, marine reptiles that, within a few million years, grew to rival the largest Paleozoic hunters. Some reached 15 meters, their bodies built for speed and pursuit. Pliosaurs with their massive jaws filled the role of apex predators. Belemnites, ancient squid relatives, gathered in dense shoals, their bullet-shaped shells now scattered through limestone beds. Each extinction left vacancies, and each time new giants emerged, shaped by the same cold, depth, and scarcity that had sculpted their predecessors. The Cenozoic era, beginning 66 million years ago, introduced a new caste. The coelacanth, once thought lost to time, survived in deep refuges, its lobe-finned form unchanged for hundreds of millions of years. Giant ostracods, armored crustaceans, settled into the abyss, their shells snapping shut against the dark. Mollusks adapted too, with some deep-sea lineages growing far larger than their shallow water relatives. These survivors reveal a thread of continuity. The deep sea, buffered from upheaval above, preserves not just ancient lineages, but ancient solutions to the same relentless challenges. Derek J. Briggs, a paleontologist who has spent decades with these fossil giants, calls this phenomenon an echo. He explains that whenever evolution faces the same set of constraints, low temperatures, limited food, and low predation, it finds the same answers again and again. The fossil record supports this. Across the Paleozoic, Mesozoic, and Cenozoic, giants return, not the same species, but the same body plans and the same strategies for surviving a world of scarcity and pressure. These patterns cross lineages and eras. Giant arthropods, cephalopods, and fish arise independently, separated by millions of years and evolutionary branches. Morphological parallels appear. Flattened bodies to resist crushing force, soft tissues supported by water pressure, senses tuned for faint chemical trails. The deep ocean acts as both refuge and crucible, forging and preserving giants shaped by the same laws. Evolution in the abyss is not random, but a memory, solutions waiting for the right conditions to rise again. Antarctica's continental shelf, swept by currents of ice-cold water, is home to some of the largest invertebrates on Earth. Here, at depths often less than 1,000 meters, pressure is modest compared to the abyss, but the water is saturated with oxygen and locked in near freezing cold. In these conditions, the familiar rules of gigantism take a different shape. The Antarctic amphipod, for example, can reach lengths of 10 centimeters, 10 times the size of its shallow water relatives. Nearby, sea spiders with leg spans wider than a dinner plate crawl across the seafloor, their bodies impossibly thin, and their limbs stretched to the edge of plausibility. These giants are not outliers, but common residents of the polar shelf. The secret is in the chemistry of cold water. As temperatures drop, the ocean's ability to hold dissolved oxygen rises. Antarctic waters, among the coldest on the planet, are also among the richest in oxygen. This abundance allows even the largest invertebrates to draw enough oxygen through their thin, sprawling bodies. For sea spiders, whose bodies are little more than a central hub for their legs, oxygen diffuses directly through the cuticle, bypassing the need for gills or lungs. The sheer surface area of their legs becomes an advantage, letting them absorb what they need from the supersaturated water. In other environments, such a design would collapse under the demands of warmer, less oxygenated seas. Here, it thrives. Low temperatures do more than slow metabolism, they extend the timeline of growth. Like their deep-sea counterparts, Antarctic giants grow slowly and live long lives. Their bodies accumulate mass over decades, not years, and the lack of large predators on the polar shelf removes the pressure to remain small and cryptic. Instead, size becomes a tool. Larger bodies store more energy, produce more offspring, and endure longer between feeding opportunities. The feast-or-famine rhythm of the deep is mirrored here in seasonal pulses of productivity, krill blooms, algal falls, and occasional carcasses swept in from the ice edge. When food is plentiful, these giants gorge. When it is scarce, they wait, burning reserves at the slow pace set by the cold. Despite the absence of crushing pressure, the Antarctic shelf produces giants that rival those of the Hadal trenches. The same convergence appears: slow growth, long lives, and bodies adapted for low-energy existence in a world of cold and scarcity. The link is clear. Oxygen-rich, frigid waters create an environment where size is not only possible, but favored. It is a pattern that repeats wherever cold and oxygen combine, whether at the bottom of the world or in the deepest trenches. Evolution, faced with the same constraints, writes the same solution into the bodies of its creatures, again and again. For most of human history, the true shape of the deep sea remained hidden. Early explorers dragged nets and trawls across the abyssal plain, pulling up battered fragments and crushed specimens, hard-shelled survivors that could withstand the violence of ascent. Soft-bodied giants, gelatinous predators, and the most elusive scavengers slipped through the mesh or disintegrated before reaching the surface. The picture these methods painted was narrow and incomplete, shaped as much by the limitations of our tools as by the reality of life below. The arrival of non-invasive cameras changed everything. In the 1990s, a quiet revolution began at the Monterey Bay Aquarium Research Institute. Bruce Robertson and his team deployed remotely operated vehicles equipped with sensitive cameras and red light illumination, technology that is nearly invisible to most deep-sea creatures. For the first time, researchers could watch without disturbing, capturing the natural behavior of giants that had always evaded the spotlight. The red glow revealed amphipods swarming in coordinated waves, isopods gorging at whale falls, and gelatinous hunters drifting with outstretched arms. What had seemed rare or even mythical became visible, their numbers and diversity far greater than trawl records ever suggested. Robertson called that moment a revelation. He warned that what scientists thought they knew about the deep sea was often a sampling bias. The animals recovered by nets were only those tough enough to survive the ascent. The new cameras showed a different reality. The giants were not exceptions, but part of the deep's ordinary order. Delicate siphonophores, colossal squid, and giant amphipods appeared in habitats once thought barren, thriving in the cold, pressurized dark. Red light systems proved especially powerful. Many deep-sea animals have eyes tuned to blue or green wavelengths, the colors that penetrate furthest through water. Red light is absorbed quickly in the upper ocean and is nearly invisible at depth. By filming in red, researchers could approach without startling their subjects. That allowed long, undisturbed observations of feeding, movement, and even reproduction. The footage captured behaviors never seen before. Amphipods arriving at whale falls in sudden massive influxes, isopods feasting until they could barely move. These discoveries forced a re-evaluation of deep-sea ecology. Patterns of abundance, competition, and survival emerged that trawls had always missed. Cameras revealed how scavengers assemble, how gelatinous predators hunt, and how slow-paced life is sustained on scarce resources. The technology changed both what scientists could observe and how they thought about life in the dark. Yet even these breakthroughs have limits. Cameras illuminate only what passes before their lens. Fast-moving predators, clever ambush hunters, and the largest, most cautious giants may still remain out of view. The ocean's vastness and the deep's impenetrable dark mean that every new technology reveals only a fraction of what exists. For every giant caught on camera, there may be others just beyond the reach of light. Each advance in imaging brings us closer, but the exploration gap persists. New systems extend range and sensitivity, from red light rigs to low light sensors and autonomous platforms, but they also remind us how little of the deep we have seen. The deep sea's true inventory remains unfinished, and our best tools are still learning how to see in the dark. This is the story of how science learned to watch giants, and how each camera lens has rewritten our sense of what is common and what is rare. It is also a warning that even with the most sensitive systems, the darkness keeps its secrets. We have learned to peer into that darkness, and still we must ask what lives just beyond our light. The ocean covers more than 70% of the planet, yet its true shape remains mostly hidden. Beneath the sunlit shallows and the rolling midwaters lies a world that is almost entirely unmapped, unvisited, and unknown. According to NOAA, only about 20% of the global ocean floor has been explored or imaged with any detail. The rest, an expanse larger than all the continents combined, remains a blank on our maps, a realm charted only by the faintest traces of sonar and the rare dives of submersibles. Within this vastness, the Hadal zone, the deepest trenches that plunge below 6,000 meters, accounts for just 1% of all ocean volume. Yet these trenches are deeper than Mount Everest is tall, and they stretch for thousands of kilometers beneath the Pacific and beyond. Their walls are steep, their floors cold and silent, and their pressure is immense. In places like the Mariana Trench, the Kermadec Trench, and the Japan Trench, the bottom is so remote that even our most advanced remotely operated vehicles have reached it only a handful of times. The rest is darkness, punctuated by the occasional echo of sonar or the faint flicker of a descending camera. OceanX expeditions have sent cameras down into these depths, capturing glimpses of creatures that seem almost impossible. Giant isopods drifting over the sediment, amphipods the size of a human hand, and the ghostly outlines of colossal squid vanishing into the black. Those moments are rare, each dive covering only a tiny fraction of the abyss. Every descent could reveal something never seen before, a new species, a new shape, a new solution to the challenges of pressure, cold, and darkness. Scientists at NOAA and MBARI speak of the deep sea as the last great frontier on Earth. The numbers are stark. 80% of the ocean is still uncharted, and the deepest trenches, the places where the giants gather, are barely scratched by human observation. In these uncharted depths, sonar sometimes returns echoes larger than any known animal, fleeting shadows that hint at something beyond the reach of our cameras. The possibility lingers that even larger giants exist, waiting in the silence, beyond the edge of what we can see. The deep ocean holds its secrets close. Each expedition, each new technology, pushes the boundary of the known outward by only a fraction. The rest remains untouched, a black expanse where the rules of life are rewritten, and where the giants of evolution may still drift, unseen, in the endless dark. Beneath our mapped continents, the deep still writes its own rules. Giant life forms thrive in darkness, shaped by forces unchanged for millions of years. With over 80% of the ocean unexplored, each new descent risks rewriting what we know about evolution itself. As technology pushes deeper, the next discovery could upend every rulebook. The giants aren't relics. They are a living question, waiting in the abyss for those willing to look. What giants will you help uncover?